So welcome to the introduction to pharmaceutical biotechnology. Okay, so as the objective, you should be able to describe the principles of pharmaceutical biotechnology in the design of biological molecules. So this is just an overview to what biopharmaceutical technology is, and then we'll have a series of lectures or recordings that are associated with respectively associated with biopharmaceutical technology. Okay, so what is biopharmaceutical technology? What is biotechnology in general? So we're saying biotechnology in general is we're saying it's a technique that comprises, or it's a technique that uses living organisms to produce or to modify products. These are drug products. Okay, so these are products that are intended for the treatment, for the management of human conditions or diseases. So we are producing drugs, and these drugs are known as biologics. These drugs are known as biopharmaceutical products. How are we producing them? We are producing them using living organisms. So we use living organisms, and hence the term bio. Technology is just a process or a system of doing something. Okay, so techniques that use living organisms, and hence the term bio, to produce or to modify drug products. What are some of the techniques then that are involved in the production of these biologics or biopharmaceutical products using living organisms? So you can use recombinant DNA technology, monoclonal antibodies, polymerase chain reaction, gene therapy, and all that as listed above. So the highlighted one is what we're going to mostly consider in this series, or in the next series, Okay, so as alluded to earlier, so we're saying biologics. This is a term that is used to refer to products of biotechnology. So all drug products, or all products that are produced by the processes of biotechnology, can be referred to as biologics or biopharmaceutical products, or biopharmaceutical drugs, okay, or biopharmaceutical medicines. So these are medicines that are produced using living organisms. What type of organisms are we talking about? So these microorganisms, you can use, or we can use, both the eukaryotic and prokaryotic cells. Apart from that, you can also use transgenic animals, tissue cultures, living cells, and cell enzymes. All these can be used to produce biopharmaceuticals or biologics, as we will see coming lectures. So the first ever, or proteins, let's just say proteins were the first novel biotechnological pharmaceuticals to ever be produced or to ever be discovered using this process of biotechnology, and specifically using the technique of recombinant DNA technology. So the first ever produced biologic was human insulin in 1982. So as we go on, you understand how it was discovered, how do you manufacture insulin, or how is insulin actually manufactured? Recombinant DNA technology. Of course, what are the advantages of biopharmaceutical technology? What do we mean? What are the advantages of using living microorganisms or living organisms to produce, to manufacture, or to modify proteins? So one, of course, they've provided treatment options for management of chronic conditions, for example, insulin dependent diabetes, rheumatoid arthritis, all autoimmune diseases, hemophilia, and all that. So they provided better treatment options for these chronic conditions, that need a lifelong or a continued supply of medications. And then also, pharmaceutical biotechnology has resulted, or has led to, the production of new potent, chemical entities, drug products, that are more effective, drug products that produce the clinical effect, a better clinical effect, at a much lower dose. So limiting the exposure of a higher dose to the patient, limiting the side effects that are associated with high doses to patients. And then why? Because it's easy to modify these proteins as they are made in vitro. So it's easy to modify them. You can modify their pharmacokinetics. I know that you can modify, let's say, their absorption, their distribution profile, so that they can become more potent. Then pharmaceutical biotechnology, that is in the third point, it has also provided, better opportunities in the development, of new approaches of diagnosis, okay, and also treatment and prevention of diseases, for example, in the manufacture of vaccines. So, like I said earlier, as we go further, we'll understand what are the different types, of diagnostic approaches, of treatment approaches, and also prevention approaches, for example, vaccines. What are the different types of vaccines that have been manufactured using or that have been formulated using biopharmaceutical technology? Okay, there's no process that is without challenges. What are the challenges that are associated with the manufacture of pharmaceutical products using biotechnology? Of course, it's expensive. Unlike the general preparation, for example, manufacturing of tablets, manufacturing of capsules, these are expensive production processes. So the production of biopharmaceutical products is very expensive because there are so many steps that are involved, in the manufacturing. So we'll see in the next lecture when we look at the process of recombinant DNA, technology as a process on its own. There are multiple steps that are involved, from the isolation to protein production and purification, making it much more expensive. And then also the challenge is that the products that are manufactured, biologics or biopharmaceutical products, they have poor absorption. Why? Because in nature, these are protein in nature, so they have poor oral absorption. Of course, this is general understanding. We all know that proteins have poor absorption. If you didn't, at least you know now. So proteins have poor oral absorption. That's why it's difficult for insulin to be administered. Insulin cannot be administered as an oral drug because of, first of all, the size of proteins of these drug proteins. They're very large, so absorption, is quite poor. Then also relative fragility. What does this mean? So proteins can easily be degraded by enzymes. Proteins can easily be affected, by the pH changes within the GIT. Okay, then we also have the common, we're saying drugs, they have to be sterilized. So common sterilization processes that are associated, with sterilizing common drugs or, common manufacturing processes to ensure the sterility of our products, they cannot easily be applied, to protein production. Why? Because, for example, if we were to use an autoclave is going to use a heat, for example, maybe 121 degrees for 15 minutes and all that, and proteins cannot withstand. Proteins, they tend to undergo thermodegradation. They are thermolabile. So sterilizing them by such processes would render them ineffective. So these are some of the challenges that are associated with the manufacture of biopharmaceutical products. And then finally, what are some of the examples of biotechnological products or biologics? What are these pharmaceutical drugs or products that have been produced, okay, that have been manufactured using biotechnology? So we have human insulin, of course, which is what the first ever produced biologic. Then also have erythropoietin, monoclonal antibodies, cytokines, interferons, and albumin. So we'll understand as we go by the different types or categories of biopharmaceutical products. Okay, and then in the next lecture, we're going to look at recombinant DNA technology as a process. So just as a way of as an overview, so in this general introduction, you should be able to just understand, what is biopharmaceutical technology? What is it? What does it involve? So, okay, so we're using living microorganisms to produce these drug products. Okay, why? What are the benefits? What are the disadvantages? What are the techniques then that are used to produce these biopharmaceutical products? You're home and dry.